Dear students, today we are going to learn about scanning electron microscope or SEM. In the last class we discussed about transmission electron microscope. There we used beam of electrons to produce a 2D image. In SEM also we use beam of electrons and will focus these electrons to the object, and variety of signals are generated from the specimen. Okay. And from that we can produce the 3D image of the specimen. Before going to the working of SEM, we can check what is going to happen if you focus the electron beam to the specimen. Suppose this is your sample and you are going to focus the electron beam here. So there will be reflection, that is backscattered electron. So these backscattered electrons, they are going to reflect back after the elastic interaction between beam and the sample. Okay, the reflections. Then there will be emission of secondary electrons. That means the electrons you focus that will hit on the electrons of the specimen atom, and these electrons they are get ejected from the orbits. So there is energy sharing and these electrons are knocked out of the sample, and this is the secondary electrons. Then X-rays, they are also produced here, that is during the emission of electrons from the sample, let it be from the inner shell, so there will be vacancy of electrons. Okay. Then the electrons from the outer shell, they can occupy these vacancies, so they will emit the excess energy as X-rays and they can move on to these vacancies. Anyway, when you direct the electrons to the sample, there will be backscattered electrons, then the emission of secondary electrons and X-rays. Okay, now we can see the different elements in the scanning electron microscope. So there is an electron gun, it produces beam of electrons, then these electrons they are accelerated to the condenser lens using the anode. This magnetic lens is called the condenser lens. This magnetic lens will focus the electron beam to the scanning coils. Then these scanning coils, they are also called as the deflection coils. By adjusting the current through the coil, we can effectively focus these electrons to the desired locations, that is to the sample. Then the backscattered electrons, the secondary electrons, then the X-rays, these are captured and analyzed. Then based on the intensity of these captured signals, we can produce the image of the specimen and it can be displayed on the monitor. So from the secondary electrons, we will get the information regarding the shape of the object. Then the backscattered electrons, it reveals the atomic number information. Then based on the X-rays, that is by analyzing the wavelength, we can predict the atomic orientation and type of the object. Okay. Now we can see the advantages of SEM. It will generate high resolution images and also reveals the atomic orientation and the chemical composition. And SEM has high magnification rate when you compare with normal microscopes. Okay. In the next class, we can study about scanning tunneling microscope or STM. Okay. Thank you.